Hi guys, this week we're going to be looking at how DNA is altered. We're going to start off with how it's naturally altered, what happens sometimes within our own bodies, and then we'll talk about how DNA can be altered in lab procedures as well. So the naturally occurring changes to our DNA are called mutations. And so the simple definition is just any change to our genetic material. But when we use the word or the term genetic material, we're talking about our DNA. So mutations are just changes to our DNA. And specifically, what's changing are the bases, those A's, T's, C's, and G's in a gene mutation. So if you have a gene for a physical trait, if you change any of those letters around, it'll change the outcome of that trait, right? So if you change a gene for hair color, change the color of your hair. If you changed a base or bases for your blood type, you could go from being type O to type B. So these changes could be in a single gene, and sometimes there's also what are called chromosomal mutations, where whole genes on the chromosome can be altered, not just one single gene, but maybe several genes, dozens of genes, or even thousands of genes that could be adjusted or affected by something happening on the chromosome as a whole. So we're going to start with the simple ones. Gene mutations really have three options. There's three possible types of changes that can be made. We call a change that happens at one location in one specific point on the gene a point mutation, right? So if there is a sequence that says A, C, C, T, G, C, and one of those bases changes, only that one point is affected, right? So when a mutation occurs or a change occurs in one specific location, we call it a point mutation, and the change could be a substitution, an insertion, or a deletion. So let's look at each of those. A substitution is the simplest and usually causes the least effect on the protein overall that is made from that gene. Substitution is hopefully what it sounds like. You substitute or you switch a base for another one. So you can see in the example down here, you have the original strand of DNA, and in red they show you a C base there, and then they show you what that DNA is transcribed into for RNA, and then the amino acid chain for the protein. Well, if you then look at the original strand compared to the mutated strand down here, they show you that that red C was switched, was substituted for a T. And that substitution causes a change in the amino acid. So if I use the codon chart and I looked up the original codon CGU, I would have found arginine or ARG. But if I look up in the codon chart what that T creates, CAU no longer codes for arginine. Now it codes for histidine or HIS, right? So it changed one of the four amino acids. Note that like the first one, methionine, and thur and lu, right, on either side, don't change. It's only that one where the mutation occurred. So that mutation is a point mutation. It happened at that one spot, and it only changed the amino acid in that one location. So again, substitutions, switching of a base, that usually results in just one single change to the protein or one piece of the protein, one amino acid, if at all, and I'll show you why in a second. Sometimes these mutations can be what are called silent mutations, where there is no change whatsoever. But for now, let's move on to another possibility. So we could switch a base called substitution. More severe changes can happen if one of the other two options were to occur. Those are insertion and deletion. And you'll see in a second why those can create a bigger change to the protein. The names hopefully are kind of giving away what happens. In an insertion, you're adding an extra base, and in a deletion, you're removing one. So let's take a look at an insertion. Here we go. Here's the original DNA strand, and they've added in red here that T. And what will happen, and the reason that they call these frame shifts, is because you read, if you're going through transcription and translation, you convert that DNA strand into RNA. That's that transcription process. And then after that, you're supposed to divide them into codons, right? Into groups of 3. Well, if you add a base in here, that means that you're going to shift or move all other bases down one spot. So notice there's an extra one left over at the end. And you shift, for example, that very first codon from AUG is now AUA. That A caused all the rest of these sequences that were already there in the original strand to just shift over one. Well, since you read them 3 bases at a time, it's now changing what those messages are going to code for. Those codons are no longer the same. So the original first codon was AUG, now it's AUA. The second was CGU, now it's GCG. Notice that it's only adding one base, but it shifts all of the other codons over so that it creates a totally different message. So notice here that it's not just one piece of the protein or one amino acid. You had MET, ARG, THR, and LU originally, now totally different. You just happen to have leucine here on the end. That doesn't change. But that's only by chance, because notice the codon UUA was the original codon. It's CUU. It just so happens that those two code for leucine. They're not actually the same, but they happen to code for the same thing. But the other 3, totally different, right? So a much bigger change to the overall amino acid code or the whole protein, right? Because each of these amino acids is part of the protein. So when you insert, you're not just changing at that one point, but you're also shifting what we call the frame of how you read the message. So same thing for deletion, just in the opposite direction, right? For this example, we're showing not just DNA, but the example if it was like sentences, right? So 3-word sentences like our 3 base codons. The original sentence would say, the fat cat ate the rat. If I just take away one letter, right? One letter, one spot. Instead of switching it, I just delete it altogether. And I still go by and separate or divide the sentence into words that are 3 letters long. My first 3-letter word is now tef, and then atta, tet, her, right? Makes no sense, sounds nothing like the original sentence. And all I did was take away one letter. So if you think of that same thing happening in a DNA strand, you take away one base, and the whole message, that whole sequence of the protein can be affected. Right? So these are called, again, frame shift, insertions or deletions. When you add or remove a base, you move everything over, you shift everything over, and it usually changes a large portion of the protein or the amino acids that make up the protein. So these gene mutations are sometimes going to cause minor changes, and usually those are the result of that substitution where you just switch a base. But frame shifts, these adding and deleting bases, cause much a bigger effect on your protein. Okay, I wanted to give you a quick peek at mutations that can result in bigger changes beyond just one gene. A chromosomal mutation, remember you have 46 chromosomes, and each of your body's chromosomes have thousands of genes on them. So if you're looking at the chromosome as a whole, you can imagine that changes to a chromosome can create some much bigger problems. So the list of possibilities are here. I just want to give you a quick description of what they would be. So here's the original chromosome, for example, much briefer, shorter, kind of simpler version of a chromosome, only showing 6 genes. Gene A, Gene B, Gene C, Gene D, Gene E, and Gene F. So each of these letters represent a gene or a piece of DNA that codes for something about you, right? So in a chromosomal mutation, you could have a deletion, just like a deletion with that gene, but instead of deleting just a base, it deletes the whole gene. So you never make that protein. Notice that B just disappeared, right? It's no longer on the chromosome. So whatever B was coding for is a protein your body will not make anymore at all. Not just a little different or a lot different. It won't make the protein at all. And then for duplication, now we have 2 B genes. And some people would go, well, that shouldn't be a problem. They're just going to make more of the protein. But what we've found is that when genes are identical, there's kind of like a genetic battle. And we have some complications when 2 genes are coding for the same thing. Translocation is a type of mutation where the gene moves locations. So here's one chromosome. Here's that original A, B, C, D, E, F, right? Here's A, B, C. Where'd D, E, F go? They switched or transferred locations to a different chromosome. Well, that makes it hard if you're a cell that's trying to create a protein and you go looking for that gene to make your protein and you can't find it, right? It's not on the chromosome it was supposed to be. It's not where it said it was going to be. Now you have to go search for it elsewhere. That's a problem as well. And same thing, inversion's kind of similar, but instead of changing chromosomes, it changes spaces or spots on the same chromosome. So you still, on this whole chromosome, have an A and a B and a C and a D and an E and an F gene. They're just not, again, where they used to be. So this is kind of a less severe version of a translocation. They're moving around, they're flipping, they're inverting, but at least they're still on the same chromosome. Over here, they've moved entire chromosomes. So again, these chromosomal mutations affect an entire gene or group of genes. It's not just moving or changing or altering one base or a couple of bases. It's affecting the entire gene. For your practice, for task 1, I'm going to ask that you do gene mutations. I want you to recognize gene mutations and see what happens to a protein when you change a base, right? So when you substitute, switch a base out, when you insert, you add a base or delete a base, what happens to that chain of amino acids? What happens to your overall protein? And just lastly, before you get started on that first task, I wanted to emphasize that when we talk about changes can be good or bad. So note, number 1, that sometimes a change is small, sometimes change is large. It's also possible that a change does not have any effect at all, right? So, for example, if we go back to that, actually, I'm going to go over to the codon wheel here, and I want to show you on this one why it is sometimes possible where a mutation occurs and the protein does not change at all. So, for example, if you have an original codon of UCC that codes for serine, if you had a substitution that changed the codon from UCC to UCA, right? So I have a switch, a substitution between C and A, I still get serine. I still have the same amino acid and therefore the same exact protein. Not just mostly the same, almost the same, the exact same. So we would call that type of mutation not just a substitution, but a silent mutation because it doesn't create any change at all. You don't notice there's any problem. The message is different, but it doesn't get any different pieces of the protein formed, right? Same thing, UCC, UCU, UCA, UCG, all of those. If any of these bases of that third piece of the codon were switched, it would not cause any change at all. And you can see that around the chart as a whole, right? UAU, UAC, tyrosine. Same thing. So if you switched a U for a C or a C for a U, no change, right? Over here, CCA, CCC, CCU, CCG, all of those, if you had any of those bases in the third spot of that codon, they'd all code for proline, right? But if I change from CCC to CAC, now I change, right? So sometimes a substitution can cause a different amino acid to be added, and sometimes it causes the exact same one. So a mutation could be silent and code for the exact same amino acid, but it also could affect change on that protein. It could create new amino acids to be added to the protein chain. So let me go back to that last slide one more time. And we say, okay, sometimes there is little or no effect, and sometimes there's genetic disorders. And this week you're going to see an example of a genetic disorder we've talked about in the past that has to do with sickle cell, and how we're taking a genetic disorder that was a result of a natural mutation, and we're going to alter it in a lab to get rid of this person's genetic disorder, to remove that disorder from their body. So we can alter genes in natural ways and also in the laboratory to remove mutations that cause negative effects. But there are also some examples of natural mutations that actually are beneficial, right? That help an organism in its daily life that help sometimes the survival of that organism. So please keep in mind that sometimes changes can be good and sometimes changes could be bad, right? If you have an organism, let's say we have a bird that lives on a certain certain location that's covered in green leaves. Well, if that bird is bright red, has feathers that are this bright red, it's going to stand out on that island, right? And if a bird stands out for predators, that's something easy to spot, and that decreases their chance of survival. Well, if that bird were to have a genetic mutation that caused its feathers to turn from being red to green, right? Now that bird can blend in. That's a beneficial, that's a helpful mutation. So changes, again, genetic disorders, examples of things that are harmful mutations. But many of the changes that occur over time, and that's what we study in our next unit in evolution, have led to helpful changes that increase the chance of survival in an organism. So there's good and there's bad. And again, option 3, sometimes neither, right? It has no effect. The protein doesn't change at all.